So I decided to do a little study over social grouping behavior of small fish in response to the presence of a larger fish. In other words, I took a group of small fish and I wanted to see how their grouping patterns would change if there was a large fish present in the same tank with them.
Under the sea, darling, it's better down
This
where
is
it's
the
west.
tank
Take it from
set
me.
up here.
Up on the shore, they work all day until the sun is slid away. While we devoting
Decorations
full
are
time to floating under the sea.
It's the mermaid and all the
Down here, all the
pink.
fish is happy as I feel my wheels a roll. The fish on the land ain't happy. They sad cause they in their bowl. But fish in the bowl is lucky. They in for a worse of fate. One day when the bugs get hungry, they soon go be on their plate. Oh oh, under the sea, under the sea. Nobody beat us, fires or eat us.
All right. So first, I felt like I should talk about the different types of grouping in fish or in aquatic animals, should I say, because I actually didn't really realize that there were differences. So a school, which is what we normally think of when we think of a group of fish, but the rules for it to be a school by definition is that all the fish have to be the same species and that the fish swim together in synchronized movements. A shoal can be a group of fish that contains different species. They're just grouped together, not all the same. And then they swim together in a loose cluster. So they're grouped together, they're swimming together, but they're not necessarily moving in sync. They're not doing coordinated movements. They're freely swimming, but together. And then a pod is what we call a herd of marine mammals. So that's our dolphins and our whales and such. But it's specifically the marine mammals. So why do fish school? What's the benefit of grouping together in a school? The advantages is it helps vigilance against predators, so being aware of their surroundings and danger. Predator assessment, so determining if a larger fish is actually a threat, because, just because larger fish is near you doesn't mean they're hungry, or doesn't mean that you're what they eat. So they have to assess whether that large fish is actually going to cause them danger. So information transfer, so basically the fish communicating with each other, them using each other's knowledge and experience and observations to stay safe as a whole group. And then predator inhibition, so basically getting the upper hand to where the predator doesn't get you. And then the physiological process. So how do fish know biologically, physiologically, how to school? And it's due to cells called neuromast cells. They're in the lateral line of the fish, and they basically sense water currents. So they can feel the movements of the other fish around them, and they're able to react based on that. They act similar to the hairs in human ears, how human ears have hairs that feel vibrations. Fish can feel the movement of the other fish around them. And something interesting I found was that studies show that schooling behavior is not a learned behavior, it's a genetic quality. So in other words, schooling fish are predisposed to school. They're not born not even knowing what a school is and then start from the ground up to get all the way to the level of being able to swim in sync. That does not happen with every generation, learning that whole process. They're born knowing how to do it. So background information regarding my study. Chemical cues are biochemical excretions of living organisms that are released into the environment and able to be sensed and interpreted by other living organisms. In other words, it's basically the chemicals that one organism excretes into the water, or their environment. In this case, it's water, and other organisms are able to pick up on those chemicals and know, hey, well, this chemical didn't come from me, and know, so they know that there's another presence with them. And then visual cues are information received by an organism through the use of the sense of vision. I feel like that's pretty self-explanatory. It's basically what an organism can see. So the overall question for this study was, are chemical cues in the environment a sufficient stimuli to elicit large social group formation in small fish? Or does the formation of large social groups require a more conspicuous stimulus? Simple terms, are fish going to be able to, or are fish going to react by forming large social groups? Are they going to react to the presence of a larger fish based on chemical cues alone, or are they going to need something a little bit more solid? So in other words, these fish have never seen the larger fish. They're going simply based off the chemical presence of these larger fish. Is that going to be enough to make them form a large social group, a large protective group? The overall hypothesis, so what do I think is going to happen overall? Subliminal environmental chemical cues released by a larger fish will be sufficient in eliciting the formation of a large social group of small fish. Additional forms of stimuli will not be necessary due to the innate predisposition of small fish to form social groups in response to the presence of a predator. So overall hypothesis, what do I think is going to happen? Chemical cues will be enough. They're not going to need any other type of stimulus or cues, such as visual cues, since the ability to form schools is something that comes natural to them. So I think that they should have some kind of natural-born intelligence, awareness of their environment for them to be able to sense a larger fish without even having to see it or anything. Just be able to pick up on those chemical cues and form a large group in order to stay safe. So, phase 1 of the experiment. It's going to focus on chemical cues and the social grouping response. So, phase 1 question, do chemical cues from a large fish encourage a group of smaller fish to participate in social grouping? Basically what I just said, are chemical cues enough? My hypothesis, chemical cues are enough, basically. So the experimental design. So first I had to figure out how to isolate the chemical cues. In other words, how could I have a large fish and a group of small fish in the same tank or at least sharing the same water, where the small fish won't see the big fish, but they can still transmit or share the small fish have access to the chemical cues released by the big fish. So in order to do that, I have an opaque, which means it's not see-through, tank divider that blocks the visual cues. And then distributing the chemical cues. So how am I going to be able to get the chemical cues from the water that the big fish is in to the water that the little fish is in? So I'm doing that based on the water's going to travel through small holes in the divider to allow the water containing the chemical cues to pass through. And then also the small fish, I had to be strategic in their placement in the tank because, we have a filter in the tank, and I'm not a chemistry fan. I don't know the science behind how the filtration works. I don't know if going through that filter would somehow alter those chemical cues or the chemical structure of those cues and change it to where it wouldn't elicit a response. So in other words, I wanted to make sure these small fish encountered these chemical cues before going through the filter in case that filter would alter them in any way. And so in order to do that, I put the small fish on the side of the tank with the filter because, and then the large fish on the side of the tank without the filter, farthest away from the filter, because the water current is going to pull the water from the side of the tank where the big fish is, through that divider, across the small fish, and then into the filter. And then the filter will release the freshly filtered water. But basically the water current is going to pull the water containing those chemical cues into the side of the tank with the small fish before it ever goes through the filter. Choosing the right subjects. This was difficult. I am not a fish expert. I do not know if one species of fish schools better than the other. You know, I had to rely on the color-coded tanks at the local fish store to tell me whether fish were, a certain species was a friendly community fish, or if it was like bad, cannot be with any other fish, or just a little bit aggressive. Based on that, I had to kind of be strategic. What I did know is I needed one large fish and then a small fish species. The small fish species must be prone to social grouping because, as we said before, studies suggest that it's not learned, so it has to be prone, it has to be genetically predispositioned to do this. And then also I had to get an effective, a good group size. So I went with 6 small fish. That's normally what they recommend, like the minimum amount to get of a schooling fish. That you need 6 of them there to really create that social group. And then control measures. I made sure I didn't rearrange the tank decor at all. I felt that manipulating the environment could cause false results because if I move a plant that they liked from one area to another area, they might start swimming differently. It just completely changes the available room that they could swim in. So my fish species. For the large fish, I went with the platinum angelfish, whose name my daughter named it Cupcake. So Cupcake. Lengthwise, they can get up to 6 inches. Height-wise, they can get up to 8 inches. Cupcake is about, I would say, probably 2.5 inches length and height. Well, height-wise, it's he or she, I don't know, is probably more like 3.5. They come from freshwater rivers in South America, and they live in temperatures between 78 and 86 degrees Fahrenheit. The small fish, I ended up going with white cloud mountain minnows. They are about 1.5 inches. They were first discovered in, I cannot see what's behind my little square. I believe it was Asia, from memory. They are actually endangered in the wild though, so I found that kind of interesting. Plenty of them on the fish market for, home aquariums, but in the wild they're endangered. And then they live in temperatures between 65 and 77 degrees Fahrenheit. And something else that I took into consideration that I didn't mention, probably more of like a control measure, is that I tried to make sure the fish that I chose had similar native temperature requirements. So I wanted to make sure that I didn't get a cold water fish and a hot water fish and put them in the same tank because just through personal experience, if they're not at their preferred temperature, they're not going to act how they normally would in the wild. And so I wanted to make sure that I was getting the most natural response that I could. So as you can see, the max for the mountain minnows and the minimum for the angelfish are about the same. And then my aquarium tank heater is set at 78. So everyone should be happy. I tried to find a happy. Oh, that needs to be gone. That video should not be there. This was a picture of the tank setup where you could see the divider in the middle, which you can kind of see it on that video anyway, so it'll be all right. All right, so phase one, the method. So day one, I put the small fish in, I let them settle in. I observe them, their grouping behavior from days 2 to 7, so about 6 days. Day 8, I put the large fish in, let it settle in, get comfortable. And then days 9 through 12, I record the small fish behavior after the large fish has been put in. I used an ethogram. An ethogram is basically a shorthand way of recording animal behavior. It's just kind of like shorthand, like a field journal, without having to write word for word sentences of what you see. You just kind of make a shorthand key and use that. So the way I did it was the number on the far left is the number of fish in the largest group present in the tank at any given time. If there's a second number, that would be the number of fish if there was a second group, so swimming independently. So let's say we had a group of 2 at the top and then 2 down here at the bottom. Those are 2 separate groups of 2. And then once you see the dash, that means that the rest of the fish, whoever isn't accounted for, they were doing their own thing. They were swimming independently. So an example, like I said, basically here I had 2 groups of 2 and then no others were grouped. That means I had 6 total. So that means I had 4 that were grouped in separate groups and then 2 that were unaccounted for. So that means that we had 2 guys just swimming around doing their own thing. The results. So here's my ethogram. And here's a chart showing the frequency of group size in white clouds. So the green represents before the predator and the blue represents after the predator. Predator meaning the angelfish. As you can see, there is very little change. We pretty much have the same number, the same occurrence of group size in the tank before and after the angelfish. However, I did notice that basically while the group size did not change, the group frequency changed. So in other words, before the angelfish, only 19% of observations without the angelfish contained more than one group of fish. That means that if we would have one group and then the rest would be swimming around doing their own thing. However, after we got the angelfish, 63% of the observations included more than one group. So that means 63% of the time I looked in and there was at least 2 groups formed. Before, it was only 19% of the time I looked in and there was 2 groups formed. So basically, to kind of interpret that, the chemical cues cause very little change in the size of the social groups. However, they did change the frequency of the social groups, and we saw a 43% increase of the number of fish who were not schooled before became schooled after. So the conclusion, there was relatively no change in the size of the social groups due to the presence of the angelfish based on chemical cues alone. However, the presence of the angelfish chemical cues elicited the urge to be involved in a group and more individuals. Phase 2, visual cues in schooling. So hypothesis number 2. I kind of built this based off hypothesis number 1. Well, kind of. So since chemical cues did not alarm the fish enough to encourage larger group sizes, schooling fish currently exposed to chemical cues that indicate the presence of a larger fish will not alter their behavior as a result of visual cues of the larger fish. Layman's terms, other words, since they already sense the chemical cues and they're not scared, they shouldn't become scared just because they see it now. So my method, the divider, what I changed up. I basically just lowered the divider 1 inch. I just pushed it further down into the aquarium gravel. I didn't change anything else in terms of like aquascaping, the landscape of the tank, the arrangement of the decor, any of that stuff. My ethogram, so basically how to read this is that we have the number on the left represents the number of minnows on the left side of the divider. The number of minnows on the right represents the number of the number on the right represents the number of minnows on the right side of the divider. And then I'm going to put this little P with a circle around it. I'm going to put that on either the left or the right, depending on which side the angelfish is on. So in other words, that documents whether the angelfish is on the left or the right side of the tank. And then here's a picture of where I lowered that divider. It's about 1 inch below the surface of the water. And that just allows the fish to still freely, it allows them to freely pass back and forth between the sides of the tank. But it also, I kept it there because we still had already established those two separate rooms per se in the tank, and so I wanted to still keep that division to see. Basically, it's also kind of testing like whether the minnows wanted to cohabitate with the angelfish or not. An example ethogram, predators on the left, we have 2 minnows on the left and then 4 minnows on the right. The results. Here's my ethogram. Basically, what we saw is that we have a chart here that shows the largest group size of minnows with a lowered divider. In other words, when I looked in, what was the largest group I saw? So the largest group size I ever saw was 6. So all 6 of my minnows were grouped up together. And the most frequent group size was 6. So most often I looked in and there were 6 of them together. And that's in response to visual cues. Position of the minnows in relation to the angelfish. Basically, 96% of the time, the angelfish and the minnows were on opposite side of the tanks. 4% of the time, that was only one of my observations, were they actually on the same side of the tank with the angelfish, with Cupcake. The conclusion, visual cues of a larger fish elicited a strong social grouping response in the small fish, and visual cues of a larger fish elicited a strong avoidance response in the small fish. In other words, the first bullet about social grouping means they were more likely to form big groups, and the avoidance bullet basically means they stayed on the other side of the divider from Cupcake. So my overall results from phase 1 and phase 2. The largest group size present in relation to the cue types. So the blue bars represent when we had only chemical cues. The green bars represent when we had chemical cues, obviously, and then the visual cues also. After the addition of visual cues, the largest group size increased from 3 fish to 6 fish. And the most frequent group size increased from 2 fish to 6 fish. Final discussion. So regarding the chemical cues only, phase 1, very little change in group size prior to and after visual cues are present. That is supposed to say chemical cues. Very little change in group size prior to and after chemical cues are present. But the frequency of the grouping behavior increased. Visual cues and chemical cues, phase 2. The group size increased, and the largest group size was the most frequent. The minnows swam on the opposite side of the divider as the angelfish, 96% of the time. So the overall conclusion, Chemical cues alone were not sufficient stimuli to encourage the formation of large social groups in smaller fish. In other words, they didn't make big groups when it was only chemical cues. However, the chemical cues did cause an increase in the frequency of small group formation. So where we saw that they were more likely to be in a group, where it went from seeing one group to seeing at least two groups every time I looked in. The addition of visual cues led to the formation of larger social groups than were formed in the presence of chemical cues alone. So basically they formed bigger groups once they could see Cupcake. And then the formation of large, I'm going to have to kind of guess what this says. I can't quite remember. Oh, okay. So the formation of large groups happened more frequently, and most frequently, once they were able to see Cupcake. So hypothesis 1 about chemical cues being enough, that was wrong. Hypothesis 2 about visual cues not really changing the behavior of them, yeah, that was wrong. And then my overall hypothesis about not needing other forms besides chemical cues, since they're innately predispositioned to form these groups. That was wrong. I was just wrong all around. So the related course material. So this is based off of an animal behavior course that I'm in, which is what fueled me to do this study. And it's basically just covers why animals learn, what animals learn, and then cooperation, which is how animals interact with each other for a mutual benefit.